Hello there, and welcome to the channel Psychology of Sleep. Tonight, we are drifting back to one of the oldest mysteries we know of. How did lifeless matter become something that can grow, adapt, and remember? Life's origins cannot be found in a single moment in time. They developed across millennia, influenced by water, the sun, and even other planets. Somewhere between chemistry and biology, the world discovered a way to come alive. Molecules began to team up, energy got captured, and tiny boundaries formed, allowing careful instructions to be passed on to future generations. What makes this story so captivating is that it is both intimate and cosmic. The atoms in you were once scattered through space. The rules that guide cells today still echo the early Earth, when the sky looked unfamiliar and the planet ran on raw chemistry. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here to one sleepy soul at a time. But for now, allow your shoulders to soften. Let your breathing slow and allow your mind to unwind as we wander through the ancient beginnings of our world. Let's begin. The early Earth had unbreathable air, and the sky was a different color. With almost no free oxygen in the air, there was no ozone shield protecting the planet. Ultraviolet light reached the surface far more strongly than it does now, and that harsh light was both a blessing and a curse. It could destroy fragile molecules, but it could also power new chemical reactions that may have fueled early life. The atmosphere was likely dominated by nitrogen and carbon dioxide, with extra gases supplied by volcanoes and meteor impacts. Depending on the mix, the sky would have been hazy rather than the clear blue it is today. It may have even changed colors due to aerosols and gases that scattered sunlight. That haze would have shaped the climate because it changes how heat is trapped and released. This matters for life's beginnings because chemistry often depends on very precise temperature windows. When you imagine the first steps toward life, try to picture an alien world, one with strange light and unfamiliar air and a surface full of molecules, reacting and competing for energy. Earth's early oceans were likely greenish from dissolved iron in water. Today, the ocean looks blue because light scatters through clear water and because tiny organisms shape what is dissolved within it. Early oceans were different. With little free oxygen around, iron could stay dissolved and travel widely through seawater. In that setting, the sea could take on a green tint, especially where iron-rich water mixed with sunlight and shallow sediments. That iron was not just a color, it was a powerful chemical player, ready to swap electrons and drive reactions. It could also interact with sulfur and other elements, building minerals that trap clues about ancient conditions. Thinking about a green ocean helps you remember that life began in a world that did not resemble postcards. The chemistry, the light, and even the palette of the planet were unfamiliar. Origins research is partly about learning to imagine that strange alien. Earth, the early sun shone more faintly, yet Earth somehow remained warm. Astronomers and geologists call this the faint young sun problem, and it sits right at the edge of life's earliest chances. With less solar energy arriving at Earth, simple models suggest oceans should have frozen far more extensively than they appear to have done. Yet ancient rocks point to flowing water, weathering, and sediments that imply long-lived liquid seas. The fascination is that the solution cannot be a single knob U-turn. It has to be a whole planetary balancing act. The mix of greenhouse gases, the behavior of clouds, and the way oceans move heat around the globe all matter. Even surface reflectivity matters because darker water and rock absorbed more warmth than bright ice. Life's earliest chemistry may have depended on this kind of subtle climate stability, where Earth managed to hold on to warmth despite a weaker sun. Greenhouse gases likely kept early oceans from freezing solid. A thickened atmospheric blanket can trap outgoing heat and keep water liquid, even under a fainter sun. Carbon dioxide is the obvious contributor because volcanoes can supply it and weathering can remove it, creating a slow planetary tug-of-war. Methane adds a more dramatic twist because it can be produced by microbes, which raises the possibility of feedback. If early life made methane, warming could increase, which supports more microbial activity and more methane production. Yet, methane can also lead to high-altitude hazes that reflect sunlight and cool the planet. So, the story may involve warming and shading and competition. What matters for origins is time. Chemistry needs long stretches of stable liquid water to build complexity. A greenhouse atmosphere, even if it fluctuated, could provide that persistence, turning oceans into reliable laboratories where reactions could run for ages without being frozen out. Life may have begun more than once, with only one lineage surviving. When conditions are right for complex chemistry, it may not happen just once. Early Earth could have hosted multiple experiments in becoming alive, running in parallel across different environments. Some experiments might have produced short-lived proto-lineages that never gained stable inheritance. Others might have been snuffed out by impacts, climate shifts, or competition. The surviving lineage would not necessarily be the first. It would be the one that became robust, adaptable, and able to spread widely. This idea changes how you feel about the common ancestor. Instead of being a singular miracle, it could be the last winner of a crowded early field. It also reflects the mystery of why life is so unified today. Unity can come from success, not from exclusivity. If one lineage developed better energy handling, better copying, or better containment, it could out-compete others and eventually dominate the planet. The traces of lost beginnings would be subtle because geology erases and because later life would recycle the same environments. The thought remains thriving. Earth may have tried many times to become alive. The oldest evidence of life is older than most visible land on Earth. The Earth was active for a long time before humans came along, and that activity erases its own past. Mountains rise, then grind. Down ocean crust forms, then sinks back into the mantle. That is why the earliest traces of biology are so hard to find. Scientists look for hints that chemistry was shaped by living processes even when obvious fossils are missing. One approach is to study how carbon appears in ancient rocks. Life tends to prefer lighter forms of carbon, and that preference can leave a recognizable pattern. The most ancient candidate signals come from rocks that have survived immense pressure and heat, which makes interpretation difficult. Still, the idea is staggering. If those signals truly reflect biology, then life was already present while Earth's surface was still being rewritten. Our planet may have hosted living chemistry remarkably early, despite impacts and a restless crust. The term quickly in geology still means tens or hundreds of millions of years. Human intuition struggles with geological time because our lives are short and our histories are recent. When scientists say life emerged quickly, they might still be describing an interval long enough for mountains to rise and vanish and for oceans to reshape coastlines many times over. This matters because it changes how you picture the first steps. You do not need a single miraculous afternoon. You have a vast span of repeated cycles, countless storms, endless tides, and uncountable chemical trials. Each day can be ordinary, yet the accumulation can be extraordinary. This kind of time also allows failure to be part of the process. Many chemical pathways can start and stop. Many protos can appear and vanish over long spans. Rare successes become inevitable because the planet keeps rolling the dice. Geological quickly is not rushed, it is persistent. It is the patience of a world that keeps offering chances quietly, over and over, until something finally takes hold and begins to evolve. Every breath you take echoes an ancient microbial innovation called respiration. Breathing feels simple, but it rests on a clever trick that microbes pioneered. Respiration is about moving electrons through a chain of helpers step by step to harvest energy efficiently. Instead of burning fuel in one burst, cells use a controlled cascade that captures energy in a usable form. Long before oxygen filled the air, microbes likely used other substances as the final electron sink, such as nitrate, sulfate, or iron compounds. Later, when oxygen became available, it offered a powerful new option because it pulls electrons strongly. That made energy yields higher, and it reshaped what life could do. Inside your cells, mitochondria still run this ancient style of chemistry. They build an electrical gradient across a membrane, then tap it like a tiny turbine. So each calm inhale links you to deep time, to microbial experiments that discovered how to turn chemistry into steady power. Early life probably swapped genes freely like a global experiment. Before evolution settled into stable lineages, genetic material may have moved sideways as easily as it moved from parent to offspring. In microbial life today, gene swapping can happen through viruses, through direct exchange, or through uptake of DNA from the environment. If early cells were simpler and lived in dense communities, those roots could have been even more common. That would make early evolution fast and inventive. A useful innovation, such as a better way to harvest energy or tolerate heat, could spread through a population without waiting for generations of descent. It also changes how we imagine the earliest family tree. Instead of clean branches, you get braided streams that join and separate. This idea helps explain why tracing deep ancestry is difficult, because the oldest signals may be mosaics, yet it also offers a hopeful message. Life may have learned by sharing, not just by competing. The early biosphere could have been a planet-wide workshop, trading successful tricks across a microbial world. Horizontal gene transfer still reshapes microbes in real time. Even now, evolution can move at the speed of sharing. Bacteria can acquire entire sets of genes that grant new abilities, like using a new food source or resisting toxins. This is not a slow change of one mutation at a time. It can be a sudden upgrade, more like installing a new toolkit in hospitals, agriculture, and natural ecosystems. This kind of transfer can change microbial communities rapidly. It also reveals something fundamental about life's origins. Genetic information is not always locked inside a single lineage. It can behave like a traveling resource that makes early evolution easier to imagine, because the first useful genetic elements could spread through communities and become refined in many hosts at once. It also complicates the search for the earliest history, because gene transfer can blur the signal of direct descent. Still, the phenomenon is a living demonstration that life is not only about inheritance through family lines. It is also about exchange across neighbors, which may have been crucial in life's earliest chapters. The first living systems probably started without any cells at all. Cells feel essential now, but early evolution may have started in a looser way. Imagine chemical reactions happening on a mineral surface or inside tiny rock pores where certain molecules keep getting remade. If a set of reactions helps create the ingredients it needs, it can persist longer than random chemistry. Add variation, add competition for resources, and selection can begin without a membrane in sight. Some researchers picture small strands of information carrying molecules like early RNA sharing space with simple catalysts. Others imagine networks that cycle and grow because energy keeps flowing through them. Only later would a true boundary become useful because it would keep helpful combinations together. In that view, cells are not the first chapter. They are an upgrade that helped successful chemistry become a stable lineage. Some modern microbes live on chemicals, not sunlight. Like Earth's earliest life, there are places on Earth where sunlight never reaches. Yet, ecosystems still thrive. At deep-sea vents and within rocks, some microbes make a living by tapping chemical energy. They may eat hydrogen, sulfur, compounds, or methane. They may use carbon dioxide as their carbon source, building their bodies from what seems like thin air. These lifestyles matter for origins because they show life does not need green plants to get started. It can run on chemistry alone. In some vent regions, microbes form the base of entire food webs in worms, clams, and other animals that rely on those microbes. That is a reminder that food can mean electrons and reactions, not just leaves and sugars. If early Earth offered steady chemical fuel, life could have taken hold in the dark long before skies cleared. Life may have begun when minerals acted like tiny chemical batteries. Picture an early shoreline or a seafloor crack where fresh, hot fluid met in not meeting place. Certain minerals can separate charges and shuffle electrons. That creates a natural voltage, like a battery you do not need to build. Rusty iron. Minerals and sulfur-rich rocks can help push carbon dioxide toward more complex molecules. The most intriguing part is that these mineral surfaces are not smooth. They are full of pores and channels which can hold on to ingredients and keep them close long enough to react. Over time, a network of reactions can start to feed on the energy difference around it. That is the kind of steady push chemistry needs. Before any genes exist to guide the work, your DNA is built from atoms forged inside long-dead stars. Hydrogen and most helium were born in the early universe, but the heavier atoms in your body took a more dramatic route. Inside stars, intense heat and pressure fuse simpler elements into new ones. Later, when some stars swell and shed their outer layers, or when massive stars explode, those newly made atoms scatter into space. Carbon, oxygen, nitrogen, phosphorus, and iron can ride outward in dust and gas, mixing into future clouds that become new stars and planets. Earth formed from that recycled material, and so did the molecules that eventually learned to copy themselves. When you think about origins, it helps to zoom out. The story begins long before Earth had oceans. It begins with cosmic furnaces and a long chain of mixing and gathering that made chemistry on this planet possible. A simple lipid bubble can form by itself in plain water. Some molecules have a split personality. One end likes water, the other end avoids it. Dropping off of these into water, and they tend to assemble on their own. They can form spheres with a thin skin, creating an inside and an outside in a single step. That boundary matters because it makes a private space where chemistry can build momentum. Early bubble walls were likely leaky, which is a feature, not a flaw. Small nutrients can slip in, while larger molecules can stay put. Even better, These bubbles can grow if more fatty molecules drift into them. Under the right conditions, physical stress can pinch them into smaller bubbles. You do not need a blueprint to get this behavior. You need the right kinds of molecules and a world that keeps stirring, drying, wetting, and mixing. The same molecule can be both food and a building block. In early chemistry, a line between eating and building can blur. A small molecule might release energy when it reacts, and that same reaction can produce pieces useful for construction. In modern cells, this principle shows up everywhere. Simple. Compounds feed pathways that make sugars, amino acids, and nucleic acid components. For origins, that dual role is exciting because it means you do not need separate systems for energy and materials. At the start, a molecule drifting in from the environment could power a reaction network while also becoming part of the growing structure. This helps explain how complexity can rise without careful planning. If useful chemistry makes more of itself and also makes sturdier products, it can gain a foothold. Over time, what began as opportunistic reactions can become organized cycles, and eventually those cycles can support inheritance. Life's chemistry may have begun as a messy mixture, not a perfect recipe. It is tempting to picture a clean, elegant pathway that leads straight to life. Real chemistry is rarely so tidy. Early Earth likely offered a cluttered soup of molecules made by heat, lightning, rock reactions, and incoming space material. Many of those molecules would interfere with each other. Some would gum up reactions, some would break down quickly. Yet, messensis can be an advantage because it creates a huge library of possibilities. If a surface, a pool, or a pore system keeps concentrating and filtering that mixture, certain reactions can start to dominate over repeated cycles of change. The chemistry that persists becomes the chemistry that matters. That is selection in a chemical form. Instead of a single lucky molecule, you might get a whole shifting community of reactions. From that crowd, patterns can emerge, and those patterns can slowly become more lifelike. Lightning can stick simple gases into amino acids in the lab. A high-energy spark can rip stable molecules apart and leave behind reactive fragments that quickly recombine. When those fragments cool and meet water, some of the products can become amino acids, which are key building blocks for proteins. The power of this idea is not that lightning is the only route, but that it proves something important. Complex life-related molecules do not require living cells to appear. Energy plus the right ingredients can be enough to start on early Earth. Sparks would have been only one source of energy in a noisy environment. Ultraviolet light, volcanic heat, and shock waves from impacts can also drive Each pathway makes a different blend of compounds, and blends create options. The deeper question becomes, which environments could collect those options, protect them, and give them repeated chances to link into larger structures? Hydrothermal vents create natural chemical gradients that can power reactions. At the seafloor, hot fluids rise from cracks in the crust, carrying dissolved chemicals from deep rock. When that fluid meets cold seawater, sharp differences appear across tiny distances. Temperature shifts, acidity shifts, and the mix of available chemicals shifts as well. Those differences matter because they provide direction. Chemistry is more likely to run when there is an imbalance to spend. In vent regions, reactions can be pushed forward again and again, not by rare luck, but by continuous flow. Many origin ideas focus on this persistence. A vent field can run experiments for thousands of years without stopping, constantly delivering fresh reactants while carrying away waste. It is a setting where energy is not a sudden flash. It is a steady pressure that can sculpt pathways over time, gradually favoring reactions that reinforce themselves. Some vents build porous rock chimneys that act like microscopic rooms. Mineralic fluids can precipitate solids as they mix with seawater, building tower-like structures that grow upward from the seafloor. Inside those structures are countless pores and thin channels which can trap molecules instead of letting them drift away. Concentration is a quiet superpower in origin research. When ingredients are held close, reactions that are unlikely in open water can become routine. The walls can also offer catalytic surfaces that encourage certain transformations. Imagine millions of tiny chambers in one chimney, each chamber running a slightly different chemical program. Most trials lead nowhere, but a few may produce compounds that feed other reactions nearby. Over time, successful chains can spread through the connected pore network. A chimney is not alive, yet it provides the kind of organized space early chemistry struggles to find. It is structure before biology and a possible stepping stone toward heredity. Warm middle ponds could concentrate molecules as water repeatedly evaporated. In a wide ocean, useful ingredients can be scattered so thinly that complex reactions stall. A small pond behaves differently because it changes with the weather. Sun warms it, rain refills it, and dry periods lower the water level until dissolved molecules become crowded together. Crowding increases the odds that two compatible molecules meet and react, especially when the shoreline leaves behind sticky films and damp sediments. When water returns, products can dissolve, mix, and travel to new microspots, which sets up the next cycle. Repetition can do what a single moment cannot. It can select for chemistry that survives drying, survives re-wetting, and keeps building. A pond also offers variety. One corner might be shallow and warm, another deep and mineral-rich, another may be shaded. That patchwork lets many chemical stories run in parallel, and some stories can join into longer ones. Freezing can also concentrate chemicals by squeezing them between ice crystals. When water freezes, the crystal lattice tends to exclude many dissolved substances. Those substances get pushed into tiny, unfrozen channels that remain liquid even in a mostly icy environment. In those narrow spaces, concentrations can rise dramatically, and molecules that rarely meet in dilute water can suddenly collide often. Cold also slows many destructive reactions, which can help fragile compounds persist long enough to participate in something more complex. This creates a useful combination: concentration plus protection. Seasonal environments are especially intriguing because they add rhythm, freeze, concentrates, and preserves. Thaw mixes and redistributes, then freezing happens again. Each cycle gives chemistry another chance to link, rearrange, and build. It also offers gentle sorting because some molecules prefer the icy interface, while others remain in the liquid pockets. In that way, winter conditions can act like a natural laboratory, nudging mixtures toward organization without any guiding hand. Meteorites carry amino acids showing complex chemistry can form in space. Some meteorites are ancient time capsules formed before Earth and preserved in the cold of space for billions of years. In certain carbonary specimens, scientists have found a surprising variety of amino acids, including types that biology barely uses. This proves that the building blocks of life are not exclusive to our planet. They are cosmic. They form in the clouds of gas and dust that eventually become stars and solar systems. When these meteorites landed on the young Earth, they may have delivered a steady supply of ingredients, helping to seed the early oceans with the tools needed for life. If a sequence of RNA formed in an environment that allowed them to persist, certain sequences could have produced shapes that helped them last longer, spread faster. Selection could then favor the strands that not only exist, but also improve their own odds of continuing. Some RNA molecules can copy parts of themselves under lab conditions. This is where the story starts to feel almost alive. In carefully designed experiments, researchers have created RNA strands that can guide the joining of smaller pieces into a complementary copy. It is not effortless, and it is not yet the open-ended replication that a living cell achieves. Still, it shows that the gap between chemistry and heredity is not an infinite chasm. The copied strand can carry information that influences what happens next, which is the seed of evolution. The struggle is accuracy. If copying is too sloppy, information dissolves into noise. If copying is too slow, the system cannot compete with breakdown. That tension pushes scientists to explore conditions that help, such as cycles of warming and cooling, or the presence of metal ions that stabilize structures. Each result is a glimpse of a possible early world where molecules began to make family lines. A world of RNA could have preceded DNA, proteins, and cells. Picture a time when the lead actor was not DNA and not proteins, but a flexible molecule that could both carry information and perform tasks. Viruses show how little you can have and still participate in evolution. A virus genome can mutate, compete, and adapt with fierce speed, yet the virus itself is not a self-sustaining system. Some scientists think viruses may preserve echoes of ancient genetic elements, pieces of early evolution that learned to travel between cells. Others see viruses as later inventions that emerged after cells existed as genetic escape artists. Either way, they force a deeper question: what should count as alive? If evolution can act on something that is only partly independent, then the border between life and non-life becomes softer, and the origin story gains proper more possible stepping stones. Some researchers think metabolism came first before genetic information. Instead of starting with a molecule that copies itself, this view starts with a network that keeps going. Imagine a set of reactions that feed one another, powered by energy from the environment. If the network uses available chemicals and produces intermediates that help it persist, it can become a kind of chemical ecosystem. Over time, such networks could become more structured, especially if they take place in a setting that provides steady inputs and removes waste. Only later would an information system become valuable because it could stabilize the best performing networks and help them spread into new places. This approach shifts the drama. It says life did not begin as a perfect replicator. It began as organized flow. Then heredity arrived as an upgrade, a way to remember successful chemistry. The appeal is that many core metabolic pathways in modern cells feel deeply rooted, as if they might trace back to the earliest energetic scaffolding that made living grow. The system's possible metabolism first ideas focus on reaction networks that self-sustain. Instead of starting with a molecule that copies itself, this approach starts with a cycle that keeps its footing. Picture a set of reactions where one product becomes the next reaction's fuel, and the chain loops back on itself. If the environment keeps supplying a few simple inputs, the loop can persist like a flame that keeps finding oxygen. The most exciting part is that such networks can grow and adapt. If a new molecule drifts in and speeds up the cycle, it becomes part of the system. This kind of survival of the persistent is a precursor to Darwinian evolution. It suggests that life's first steps were not about information, but about endurance. Gradients are attractive for origin theories because nature can create them without biology. Vents can do it. Rock chemistry can do it. Even simple membranes can hold a difference for a while. Once a protocell could maintain any gradient at all, it would gain a steady source of usable energy which could support longer chains, better catalysts, eventually more reliable inheritance. The universal genetic code is shared by almost all known life. Every cell translates genetic instructions into proteins using the same basic dictionary where three-letter nucleotide words map to amino acids. That shared system is so widespread that it is one of the strongest clues that life on Earth is deeply related. It is also a strange kind of fossil. The code is not the only possible code. Many alternatives could work in principle. Yet, evolution settled on one and then built everything else on top of it. Once the code became central, changing it would be risky because every protein would be affected at once. That makes the code feel like an early lock-in event, a choice made when life was still young and flexible. The rare exceptions and small variations we see today are interesting precisely because they are limited. They show that the code can bend a little, but it usually does not break. That stubborn consistency is part of what makes origins feel real, rather than mythical. That shared code suggests all life today has a common ancestor. When you see the same translation system in bacteria, plants, and animals, it implies a single deep lineage behind today's diversity. This does not mean evolution had a neat starting point with one perfect cell. It means that at some stage, one family of early life became so successful that its descendants filled the planet and outlasted competitors. The common ancestor idea is powerful because it narrows the mystery. Whatever life's first steps were, they led into a lineage that learned how to store information, translate it into working chemistry, and pass improvements forward. It also means you carry an ancient inheritance that predates bones, forests, and even oxygen-rich skies. The details of that ancestor remain hidden because early life left few clear fossils, and because gene swapping can blur family trees still. The shared code is a signature that points backward toward a single route somewhere in deep time. The last universal common ancestor was likely a community, not one cell. It is tempting to imagine one tiny organism standing alone at the dawn of life. Many scientists suspect the reality was more like a bustling microbial neighborhood. Early cells may have been simpler and leakier, and they may have shared useful molecules with their surroundings. If genes moved easily between neighbors, then innovation could spread quickly, and the ancestor becomes less like a single individual and more like a population disease that collectively explored solutions. This community view also helps explain why some core features are universal, while other features vary widely. A shared pool of genetic parts could be mixed and matched, with successful combinations persisting. In that era, boundaries between lineages may have been fuzzy, closer to a network than a tree. Over time, as membranes improved and genomes became more coordinated, lineages could become more distinct. The last universal common ancestor in this picture is a shared chapter of communal evolution rather than one lonely cell. The earliest ecosystems were microbial mats, layered like living carpets. Long before animals swam or plants rooted into soil, microbes formed dense communities across shallow waters and wet surfaces. A microbial mat can be layered with different organisms living in different zones based on light, oxygen, and chemistry. The top layer may catch sunlight. Lower layers may rely on chemical reactions that use the byproducts from above. This creates a miniature ecosystem in a few centimes that change over the course of a day. Mats can also trap sediments, bind grains together, and gradually build structures that can become rock-like over time. What makes mats so captivating is their cooperative architecture. They show how simple organisms can build complex habitats just by living side by side. For origins and early evolution, mats offer a plausible stage where chemistry, energy, capture, and recycling could intensify. They concentrate life, they create microenvironments, and they preserve traces that can survive into the geological record. Stromatolites are rock-like structures built by microbes over time. Layer by layer, microbial communities can trap sediment and bind tiny grains together, slowly making a structure that looks more like stone than life. The builders are often microbes that live in dense sheets where the surface is active and the deeper layers are sheltered. As water flows over them, grains settle and stick. Over days and seasons, those grains become part of the mat's growing architecture. The result can rise into domes, ridges, or columns depending on currents and sunlight. What makes this astonishing is the timescale. A living film can create a lasting geological signature, one quiet day at a time. These structures show that early life did not need claws or shells to leave a mark. It could shape landscapes with patience, turning metabolism into architecture and turning a shoreline into a record keeper. Some stromatolites are living today, offering a window into deep time. In a few places on Earth, stromatolite communities still grow, which means you can stand beside a modern ecosystem that resembles some of the planet's earliest ones. These living sites are rare because most environments are now crowded with grazers and competing organisms that can break mats apart. Where conditions are challenging, such as very salty lagoons, microbial layers can still dominate. That rarity is part of the appeal. It highlights how different the early world was when microbes had vast space and little interference from larger life. Studying living stromatolites helps scientists connect biology to geology in a direct way. You can observe how layers form, how sediment becomes trapped, and how microbial activity changes the chemistry around it. It is not a perfect time machine because today's Earth is different. Still, it is a tangible reminder that ancient styles of life never fully vanished. They persisted in pockets, quietly continuing a tradition older than animals. Photosynthesis began without oxygen, using other chemicals as electron sources. The first photosynthesizers likely did not split water. Instead, they used hydrogen, sulfur, or iron to fuel their growth. This was an incredible step toward independence. Instead of relying on rare chemicals from vents or meteorites, life learned to harvest energy directly from the sun. Later, a lineage emerged that could use water as its electron source, that released oxygen as a waste product, a change that would eventually rewrite the entire atmosphere and pave the way for complex animals. But it started quietly in the shallow, sunlit corners of a young world. Nitrogen fixation is an expensive chemical feat that life mastered early. Nitrogen is everywhere in the air, but it is locked in a form that is incredibly hard to break apart. Life needs nitrogen to build proteins and DNA, so early organisms had to find a way to fix it into a usable form. This requires a massive amount of energy, and the machinery is sensitive. That means a fixer must balance expense and benefit, and Many organisms solve it through partnerships. Some bacteria live alongside plants, exchanging fixed nitrogen for sugars. In origin stories, this kind of chemistry suggests a major evolutionary leap where life learned not just to adapt to its environment, but to unlock a global resource and make it biologically accessible. That enzyme uses metal clusters like tiny industrial machines. At the heart of nitrogen fixation sits a protein complex that relies on tightly arranged metal atoms, often including iron and sometimes molybdenum. More than these metals are not decoration. They create an active center where electrons can be handed off with precision and where nitrogen can be coaxed into new arrangements. This is another example of life using the planet's raw minerals to perform its most essential tasks. Defining life is tricky when chemistry starts to organize. At the start, the boundary between living and non-living can look more like a gradient than a line. A crystal can grow, yet it is not alive. A fire spreads, yet it has no heredity. A chemical network might sustain itself for a while, concentrate its own ingredients, and respond to changes, yet still fall short of what we call life. Origins research forces you to ask, what do you really mean by living? Is it metabolism, a steady use of energy to maintain structure? Is it compartments, a protected inside with its own chemistry? Is it information stored patterns that can be copied? Or is it evolution, where improvements accumulate over generations? Different definitions highlight different milestones, and each milestone suggests different environments that could support it. The fascination is that life may not have arrived in one leap. It may have arrived as a sequence of achievements, each one making the next easier. In that view, the earliest stage might not look like a tiny cell. It might look like chemistry that has started to behave with purpose, even though no mind is present. Some researchers use Darwinian evolution as the key threshold. One useful way to draw the line is to ask a simple question. Can a system evolve through selection where variations compete and the winners become more common? If the answer is yes, then the system can improve over time without needing a designer. A neighboring protocell with less effective chemistry may shrink, leak, or fail to reproduce. This creates a clean stage for competition where success is linked to a physical container. Compartments also allow experimentation. Each tiny bubble can run a slightly different chemical program, and the environment can sort them by performance. Some will be dead ends, others will persist long enough to be improved. Once you have that, evolution has a handle. It can favor systems that keep useful combinations together, which is a major step toward the organized complexity we associate with living cells. Early life fed on geochemical energy from Earth's interior. Before food, webs, and oxygen, a young biosphere would still need a dependable energy supply. Earth's interior provides one through chemical disequilibria, where reduced compounds from deep rock meet more oxidized conditions near the surface. When those chemical differences interact, energy is released, and that energy can be captured by metabolism. Think of it as a planet that is still cooling and reacting, offering fuel lines that do not depend on sunlight. Such energy sources can be steady over long times, especially in regions with active circulation of water through crust. This matters for origins because steady energy supports persistence. It allows reaction networks to keep running, protocells to keep cycling, and small improvements to matter. It also makes the search for life beyond Earth more interesting. If a world has water and rock interactions, it may have energy even under ice or in darkness. Life's earliest diet may have been the planet itself, served through chemistry. Today, deep subsurface microbes still live, beneath ground. Far below your feet, there are organisms living in cracks, pores, and thin films of water. In places without sunlight and with painfully limited nutrients, some survive on slow chemical reactions between water and rock, and they can persist at rates that make surface life seem frantic. Their existence expands what we consider a habitable world. It also offers a living analogy for early Earth, when surface conditions could be unstable and protective niches would be valuable. In the subsurface, temperatures rise, pressures increase, and the environment is buffered against rapid change. That stability can support long-term survival, even when growth is slow. Deep microbes also reshape geology in subtle ways, altering minerals and influencing the chemistry of groundwater. For origins thinking, this matters because it suggests life can occupy hidden spaces for immense stretches of time, quietly accumulating adaptations. If early life ever retreated underground during harsh surface periods, it could have endured, evolved, and later re-emerged to repopulate more welcoming, habitats. Some ecosystems rely on hydrogen made when rocks react with water. In certain environments, hydrogen is produced naturally when water interacts with specific minerals in the crust. That hydrogen can become the energy backbone of an ecosystem, feeding microbes that combine it with carbon compounds and turn that energy into growth. What is striking is the independence of this food chain. It does not begin with sunlight. It begins with geology. In vent regions within oceanic crust and in some continental settings, hydrogen can seep into surrounding waters, and microbial communities can gather bio where the supply is strongest. These organisms are often masters of scarcity, extracting energy from reactions that seem modest yet are dependable. This kind of ecosystem is exciting for origins and for astrobiology because it shows a pathway that can operate in darkness, under ice, or deep underground. If a planet or moon offers water-rock reactions that generate hydrogen, it may also offer a foothold for life. Hydrogen becomes a whisper of possibility carried by stone and water. Sepentization can produce hydrogen and simple organics naturally. When certain mantle-derived rocks react with water, their minerals change structure and chemistry. This process, called serpentinization, releases hydrogen as a byproduct, and it can also support the formation of small organic molecules under the right conditions. The fascination is that it links planetary geology to lie-like chemistry without requiring biology to start the chain. The reactions can also raise the alkalinity of surrounding fluids, creating distinctive chemical environments that persist as long as water continues to be circulate through rock. Over time, serpentinization can build a steady chemical landscape with energy-rich molecules and mineral surfaces that can catalyze further reactions. This makes it a favorite mechanism in origins discussions because it is not a rare event. It is a repeating geochemical engine. It also appears to be plausible on other worlds with water and rocky interiors. In that sense, serpentinization is not just an Earth story. It is a planetary recipe that could run in many places quietly, producing fuel and starting materials in the dark. Alkaline vents create conditions that resemble parts of cell chemistry. In some vent systems, fluids are strongly alkaline, and they meet relatively more acidic seawater across mineral structures. This sets up differences in pH and chemistry that can be maintained for long periods. What makes this so tantalizing is how closely it echoes a core strategy used by cells, which is harvesting energy from gradients across boundaries. In an alkaline vent setting, the boundary is mineral rather than biological. Yet, it can still separate environments and guide ion movement. The mineral pores can also concentrate molecules and provide catalytic surfaces, which helps reactions proceed in a more organized way. This combination, steady gradients plus structured microspaces, gives origins researchers a coherent stage for early metabolism like processes. It offers a way to get direction, persistence, and concentration without needing advanced enzymes or complex membranes at the start. The vent becomes a natural reactor with built-in architecture where chemistry can be driven repeatedly toward greater complexity. The boundary between rock and water may have life's first home. Interfaces are where chemistry gets interested. In open water, molecules drift apart. On a rock surface, molecules can stick, concentrate, and meet again and again. The rock can also create microenvironments, small pockets with different acidity, different ion balance, and different access to catalysts. Water brings mobility and mixing, while rock brings structure and stability. Together, they create a setting where reactions can be both repeated and shaped. Many origin scenarios, even when they disagree on details, return to this meeting point. It is a place where energy gradients can exist, where minerals can assist transformations, and where compartments can begin as pores or films before becoming true membranes. It is also a place that exists in countless forms, from seafloor cracks to shoreline sediments to hot spring terraces. If life began at an interface, it began in a place where the planet itself could guide chemistry, not by intention, but by providing surfaces, cycles, persistent boundaries that make complex reactions more likely to take hold. Life's earliest fossils are subtle, and scientists argue about some claims. The oldest traces of biology are rarely neat bones or shells. They are whispers in stone, faint textures, tiny shapes, or chemical patterns that might have many explanations. Heat and pressure can squeeze and distort ancient rocks, and flowing fluids can rewrite them from the inside. That makes early evidence feel like a crime scene that has been cleaned up by time. Researchers look for multiple clues that agree, such as microscopic structures that sit in the right setting, paired with chemistry that fits biological activity. Even then, debate is healthy because geology can be an excellent imitator. The most exciting part is that disagreement does not mean failure. It means the questions are sharp. Each contested claim pushes new methods, better imaging, and stricter standards. When a signal survives that scrutiny, it becomes more than an interesting idea. It becomes a rare, hard-won window into Earth's deepest biological past. Carbon isotopes can hint at biology, but geology can mimic them. Carbon comes in slightly different forms, and life often favors the lighter version because it is easier to process in many reactions. That preference can leave a measurable fingerprint in ancient rocks. It is like finding a pattern that suggests a familiar hand was at work. The catch is that non-living processes can sometimes create similar patterns depending on temperature, pressure, and the history of fluids moving through rock. That is why carbon isotope evidence is powerful, yet never enough on its own. Scientists try to link isotope signals to context. They ask what kind of rock it is, how it formed, and whether it has been altered since its birth. They compare the signal with other chemical indicators and with the microscopic fabric of the sample. When multiple lines of evidence align, isotopes become thrilling. They can suggest life's presence even when no clear fossil shape exists. They can also reveal how early metabolisms may have processed Carbon in a world unlike today. Micros' fossils can be faked by minerals, so confirmation needs many tests. A tiny filament in rock can look like a cell chain. Yet minerals can grow into similar shapes through purely chemical processes. Crystal growth can produce tubes, spheres, and branching forms that seem biological at first glance. That is why a single photograph, no matter how compelling, cannot settle the question. Researchers treat candidate microfossils like fragile evidence that must survive cross-examination. They examine the surrounding rock layers to see if the environment makes biological sense. They analyze chemistry to check for carbon where it should be, and for minerals that form in ways consistent with life. They use multiple imaging techniques to see whether the structure is hollow, layered, or composed of mineral grains that signal a non-living origin. The goal is not to dismiss exciting finds. It is to protect the story from wishful thinking. Each rigorous test brings us closer to a true memory of life's beginning. Along the way, we saw how life borrowed from its It used minerals, gradients, metals, and cycles that the planet already offered. Nothing appeared fully formed. Everything built slowly, layering success upon success until chemistry became biology, and biology became a living history that still flows through you now. You might imagine those early environments settling into the dark, the slow movement of water through rock, the steady rhythm of tides, the warmth of vents, and the long patience of time itself. Let that vastness soften your thoughts. You do not need to hold every detail. The story can drift like sediment settling after a long current. If you find yourself still awake and curious, there is another video waiting for you on the screen, ready to carry you into a new quiet exploration. And if these nighttime journeys have brought you comfort or wonder, you are always welcome to support the channel by liking, subscribing, or sharing a thought. It helps these stories reach others who may need them. For now, allow your breathing to slow. Let your body rest where it is. You have traced the path from the ancient to the now. Rest well.